Hello, this is Russ Bainer. I'm a senior data scientist at Maza Therapeutics in South San Francisco, California. And I am very excited to be able to give a talk on the foundations of data science for bioinformatics, as I see it, at least. Before I begin, I want to thank the organizers for giving me the opportunity to talk a little bit about my work and the things that I think are most useful to early career scientists. And as you can see, there's my contact information down at the bottom of the screen. So the organizers suggested that I focus my talk on more fundamental principles of data science and the kinds of things that I think might be useful to early career scientists as they consider launching into the data science and bioinformatics space. So what follows is my interpretation of that. So I've decided to structure my talk like this. There will be three parts. First, I'm going to talk a little bit about what bioinformaticists actually do all day, because I think depending on where you are in your career, it might not be obvious that what functionally happens and what a bioinformaticist would really do. Then I'm going to spend a good chunk of my talk describing how bioinformaticists actually do their job and how they specifically provide value to the enterprise of modern science. And I'll give you some ideas about what I think is most important about the bioinformatics role there. And then finally, I'll wrap up with a little bit of just early career advice about key skills and techniques that I think are worth learning, and kind of the way that I think it's best to think about developing a career in bioinformatics, should you choose to do that. Okay, so without further ado, I'll start. I thought I would begin by talking a little bit about the company that I work for, because I think that Maza Therapeutics is actually a really great example of how applied data science and biotechnology really can improve the way that we think about drug discovery research and ultimately, hopefully, provide solutions to really bad disease problems. By way of background, Maza is a small startup that was launched in 2019. Right now, we're at about 80 employees, mostly localized in South San Francisco, although obviously with the current situation around the pandemic, many of us are working remotely, and that's why you hear me as a disembodied voice. The core concept that we follow is something related to understanding how genetic modifiers could lead to new therapeutic modalities and help people who are sick stop being sick. So the intuition there, for those of you who don't really know very much about genetic modifiers, is that there's this kind of fundamental question about the way that the human genome actually manifests as diseases or not diseases. So as you know, we live in a world where we're able to sequence an entire genome of a person, and we're even able to, through very careful analysis and scientific work, identify, for example, the gene that seems primarily responsible for a person developing a disease or a condition. Now, that said, it's also very clear that even though we believe we understand the underlying mechanism that drives a disease very well, there are clearly individuals that have a disease-causing mutation in a gene that is unambiguously responsible for a disease, but nevertheless don't get sick. And this has been really shown very clearly back in 2016, where the Resilience Project was published, in which the authors identified a large group of individuals who they believed on the basis of their genomes should really have a very serious childhood disease, but didn't. And so the founders of our company asked why. So it's broadly known generally that there are genes that can protect people from certain diseases, but they were also curious about whether you could standardize this into a genetic platform where you could identify these so-called genetic modifiers that protect people from illness in a more standardized way and identify useful therapeutic targets. So around the time of that report, one of the Maze founders, Jonathan Weissman, in his lab was developing novel applications of the CRISPR technology, which you might know received the Nobel Prize this year, that was related to understanding gene-gene interactions, that was focused on understanding how genes interact with each other. And at the same time, Steve Elledge, who won sort of the US equivalent of the Nobel Prize for his work in DNA damage repair, was also interested in applying these advanced functional genomics tools to study these interactions. So they were initially focused on understanding how these interactions could be used to kill cancer cells, but the interesting thing that Jonathan in particular discovered when he was looking for these synthetic interactions was that there were also combinations that appeared to be protective, that his lab described as having something called a buffering effect. So the idea then was to turn this insight about identifying these buffering effects that essentially confer protection against potentially horrible diseases into a full company, was essentially the genesis of Maze. So the early Maze team started to build this company, and they had this idea that they could identify naturally protective variants from human genetic data. And then we could generate some functional genomics data that complements those population level insights to identify modifiers that could be targeted with new therapies. And then those therapies would therefore be useful in populations that have a genetically designed disease. And at the same time, we got sort of a surprising boost in confidence about this whole approach, because there was this drug approved in 2016 based on this exact idea for a pretty horrible neuromuscular disease called spinal muscular atrophy. So on the basis of a very large body of work, that drug was designed to increase SMN2 protein levels, which was found to help patients with this SMN1 disease-causing mutation, and which allowed the SMN2 protein to compensate for SMN1 and essentially prevent the disease from manifesting in people who really should otherwise have it, absent activity in SMN2. So with this example in mind, our goal was to build a platform that could systematically identify these drug and genetic modifiers for severe genetic diseases. So the approach was to develop this purpose-built platform that integrates high-value human genetic and functional genomic data from public and commercial and proprietary sources. So that includes things like the UK Biobank and Open Targets, and various other publicly available data that anybody can see. It also involves more proprietary sets like FinGen. I personally am a advisor on the H3 Africa project, and as well as proprietary cohort data that is generated in-house in particularly valuable and interesting populations, where we think we can get some proprietary genetic insights. And then the idea is that once you've amassed this critical body of evidence, you can perform a series of experiments that interact with one another, including genome-wide CRISPR screens, mutational scanning experiments, interactomics experiments, and various other sort of traditional biological approaches. You can form some sort of comprehensive integrated way of interpreting those experiments together, and then more efficiently prosecute drug discovery. So identify a compound that a patient who has a horrible disease can take and then not be as sick as they were before. So that's kind of the core mission of this company. Now that you understand how Maze was founded and what the centralized focus of this company is, I thought that I would unpack a little bit about how bioinformaticists really contribute substantially to this mission. So you can probably guess from that description of this heavily integrated network of experimental and data sources that are going to be comprehensively combined to generate insight. There's a lot of computational work to be done, and also just processing and organizational work that sits squarely on the shoulders of bioinformaticists. So I thought I would begin by talking in a sort of abstract way about exactly how an experiment tends to work from the perspective of a bioinformaticist and or data scientist, and kind of help develop some intuition about what our core job is. So as you all know, experiments typically begin with some sort of experimental question that's of interest to your drug discovery program or whatever program it is that you're focused on. On the basis of that experimental question, you're going to develop an experiment that allows you to collect data that's pertinent to understanding understanding something a little bit more about the question that you have. So once you have all that raw data in hand from whatever your experimental system is, then typically there's a pre-processing step, or maybe several pre-processing steps in which those data are converted into a format that's more interpretable by humans, for example. Then there's usually a series of quality control steps in which you basically use internal metrics to determine whether this primary experiment was successfully completed. There's usually also a potentially long formatting process in which once you're satisfied that that process data does actually potentially contain biological insight, you now need to get it into a format where a human could fit some sort of statistical model on it, or possibly just otherwise understand it a little bit better. And then once those data are formatted properly, then you have an actual analysis where those data are communicated to usually a scientist or a data scientist who actually performs some sort of operation on that prepared data. So identifies genes that are differentially expressed, identifies constructs that cause cells to die, that kind of thing. And then on the basis of those sort of mechanical operations, ideally you develop some sort of insight about what the underlying biological process is that you're studying. Okay, so this is a fairly long and complicated system that hopefully results in insight that allows you to generate a new experimental question. And you can probably also appreciate that as technology advances, these experiments get much more complex, the pre-processing steps get much more complex and nuanced, the QC becomes much more complex, et cetera, et cetera, et cetera, right down the line. Basically just introducing a huge amount of difficulty for a human to understand exactly what happened when they performed their experiment. And the way that I like to think about it is that there's this kind of gradient of complexity and informativeness that you're producing through this process. So when you have that raw data, it's not very complex. It might just be a bunch of A's, C's, T's, and G's. It might be CT curves. It might be some measurement of the amount of light coming off of a fluorophore, but it doesn't really tell you very much by itself. And through the work of these different steps, you're kind of creating more and more complexity to your understanding of what happened in the experiment. And hopefully you're also increasing the informativeness of that experiment to a scientist. But in practice, what the real challenge is that as you add that complexity, you usually have to continually make new decisions about what the appropriate path forward is to move data from the QC step into the formatting step, into the analysis step, et cetera, which means that you really can't standardize things as easily once you get into these more complex parts of your experiment, okay? So typically you can standardize early things like pre-processing fairly well because you always do it the same way to every data set that comes in. And so you can program a computer to just take a sequencing read and turn it into, for example, a location within the human genome. because there's just one way that you do that, and you do it the same way every time. But as you get up to these harder and harder parts of the experiment, you really can't standardize and reuse things as easily, which means that there's this kind of roadblock to the amount of work that an analyst or a bioinformaticist actually needs to do. And the key goal at Maze is to kind of change the way that this works, so that basically what you do is you increase that standardization and reusability throughout more and more of the early stages of this pipeline, such that you have a kind of standard practice that you go through. It's heavily tracked and recorded, and ideally you can get to a relatively complex and informative location fairly quickly, on the basis of essentially standardized tools. So what that does in practice is essentially lop off a huge amount of the work that a scientist would actually have to do, so that they can spend their time really thinking about the hard questions that arise from a good experiment. Okay, so that was a very abstract way of talking about this problem. In a more practical way, you might think about, okay, first you've got this experiment that you've performed, and here it's a picture of cells in a dish. This could be a sequencing experiment. This could be measuring the tides outside of your house and writing down the numbers of how high and how low they get every day. It could be any kind of data collection exercise. But again, it turns into an analysis where you take the numbers that you've gotten off of your computer or off of the stick in the water or whatever, and turn those into some sort of value, that a statistic that represents the evidence for a change or some other important thing about those values that you're trying to measure. And then on the basis of that analysis, you might create an interpretation of what those numbers mean. So in this example, I'm saying, okay, maybe you're learning something about where a small molecule binds to a protein, but it really could be any sort of insight that you wanted to develop. So that's at least like the kind of the bare bones structure of how an analysis would work, that would provide more information about a question that you're asking. But in a modern bioinformatics or a biotechnology milieu, what you actually get is a whole series of experiments that are all kind of conceptually related and that all inform each other in the analytical steps. So the way you can imagine this is that every analysis might actually draw information from lots of different experiments in a way that's fairly complicated, and where the information that you gain from each one of them provides you with a different aspect of the raw data that, to a smart analyst, will allow you to answer a fairly small question. And then those small questions kind of all line up with each other to provide information about a much more complicated question that you're trying to resolve. And so just by looking at this picture, you can see that suddenly things become pretty complicated pretty quickly, because you have experiments that provide insights to an analysis, but also that analysis maybe changes the way that you do an experiment, and then that experiment is also informing another analysis. And I haven't even drawn them, but there's probably also interactions between these analyses themselves, and that those analyses nevertheless need to be reconciled with one another to come up with a kind of comprehensive picture about this idea. So this is really where data science enters into this whole enterprise, is through clarifying and organizing those data and providing automation to the parts of that analysis that really the person who's thinking about the problem shouldn't need to think about. So what I would argue is that a key role of bioinformatics and data science is essentially to identify these arrows, these places where either an experiment informs an analysis or an analysis informs an interpretation, and figure out how they can be automated. So if you have an automated way to go from, for example, the CT values on a PCR reaction into an estimate of how much a gene's expression level changes in an experiment, then basically you can forget about that connection, okay? And when you're an analyst, you can start to think about just the gene expression level change as the relevant unit of information, and not really all of the steps that go from the way that you handled those cells in the dish to figuring out how much that gene's expression level changes. And similarly, you can abstract away other inferences. For example, maybe this is an experiment where you compare reagents against each other, and you decide, oh, this compound influences my cells better than another compound. You can then abstract that away, turn that into a unit of information that's just available to the analyst, and prevents the analyst from going back to this experiment, trying to sort out what the best choice is, and following all of the kind of logical steps that provide that insight. Now, data science is very focused on identifying and getting rid of these errors, and I would argue that in general, we're pretty good usually at getting rid of these arrows on this side. So more and more people are good at standardizing the way that raw experimental data turns into something that an analyst could really think about. But one of the things that's really hard to do is figuring out how to codify the relationships between these analyses, and also the way that those analyses are integrated into a comprehensive idea of a biological system. And that's a core problem that Maza is really trying to solve right now. So this is the way that Maza actually approaches that problem. So first of all, you can think about the experiment from the perspective of a data scientist. So now I'm not talking about moving cells around in a dish. I'm really talking about the way that the data actually enters into the organization. So typically what will happen is we will get raw data from somewhere, either our internal instruments from experiments that we've done. We might buy data from a vendor, so somebody else does an experiment on our behalf and we get the data from them. Or there might be an external source, for example, the UK Biobank or another big consortium might just publish their data online, and all of those things would just be essentially deposited in data storage. From that, we have a suite of what we call pipeline tools that are held in something called a Git repo. Now, if you don't know what that is, that's okay. But you can think about this as basically a repository that contains all of the software that I and other data scientists at Maze have written that is meant to take the raw data that's been deposited in the data storage location and process it in a very standardized way. So for example, you might take a sequencing data set from an instrument. So this would be long sequences of A's, C's, T's, and G's that are maybe derived from a genome somewhere. And you might have a tool that aligns them to a genome and turns them into a new data structure that represents the locations of in the human genome that match the sequences in that initial file. Okay, and then the key insight is that these pipeline tools are highly iterative. So you might then have another tool that takes the thing that process data structure from your first tool and is able to convert those locations in the genome into, for example, a count matrix where you can ask, okay, this is how much a gene is expressed or not expressed in a given sample. So there's a whole thicket of these tools that are all interacting with each other in a very automated way, and they all pass information to each other about what came in, what went out, and the various inputs and outputs and relationships between the things that went into the tool and the things that it produces. Now, at some point, you get to these automatically processed data structures, and you've really kind of hit the wall, and you can't automatically improve them in a way that will provide more insight to a human. So then we transition into the analysis phase. And the way this works is an analyst, so that would be me or one of my colleagues, would sit at a computer. They would import those processed data structures in a very controlled way, where we keep track of everything that is coming in to the analyst computer implicitly. They would generate usually some sort of code that would interact with this processed data structures, which is again saved in one of these repositories, which really just means that we are able to, which really just means that we're able to, which really just means that we're able to all see the work that the others have done, and we can see how we've changed our code and what decisions we all made. That analyst will then creates something, usually a report or an image or a result object, which then gets registered into a results database. And then once something makes it into here, people say, okay, this is a real result. A human has thought about it. Our processes have recorded everything that we care to record about it. And now it's that result is available either to users to look at directly. So you could go to a website and say, here's the picture that Russ created in his report. Or we also have a series of platform apps which automatically ingest the results objects produced by an analyst and allow us to borrow information across experiments to gain other insights. So an example of one of those platform apps would be, for example, an aggregator of signals across similar experiments. So in this example, you might imagine that in our results database, we have data results that have been created by analysts for 3 different programs, for example. They're all focused on different biological problems. And they all contain lots of data derived from lots of different experiments. So you might have a bunch of gene expression changing experiments. You might have a bunch of experiments that involve screens of some kind. You might have human genetics data. These apps are able to essentially crawl all over this database, identify particular kinds of experiments that can be unified in a kind of comprehensive way. And for example, in this tool example, you might be interested in what happens to this gene when you perform a CRISPR screen and how does it affect enrichment and depletion in all the different screens that you've done throughout your organization. And you might have rich information about all of these. So maybe you know that these are in one cell type and these are in another cell type, and you know that these are treated with a drug and these are just allowed to proliferate over time. And by looking at what happens to this gene across all of these different experiments, you might be able to derive something more comprehensive. So maybe, okay, this experiment, and this experiment, they're all done in the same cell line, and there's some known property about that cell line that makes it so that we can conclude something about the activity of this gene just based on the fact that it's always enriching across all of these experiments. Maybe these are 2 experiments that are done in cell lines that have mTOR signaling active or something like that. And that would potentially provide some other insight. So to kind of wrap up this part of the talk, again, the way this actually looks to a scientist outside of the data science realm is that the scientists produce data through experiments that goes into our data storage. Then we have these automated processing pipelines, which allow us to do a lot of the early work automatically in a way that's tracked and understood for downstream users. Then we have another system for tracking the analysis choices of a computational biologist or a bioinformaticist. And then we have a results database where everything gets registered and we learn everything afterwards. And from that, downstream users can hopefully learn something about the consequences of the experiment. And importantly, because we have networked together all of these experimental insights, we can implicitly gather information that is potentially pertinent to other experiments by borrowing across different comprehensively tracked enterprises. Okay, so with the last few minutes, I want to say I want to say a little bit about what I think it takes to be a good bioinformaticist. So if you're at an early stage in your career, you have to make a lot of decisions that actually end up having a lot of core implications for you and your life. The first thing that I would say is that to be a good data scientist in general, the main thing that you need to do is learn how to be a good scientist. So what that means is figure out how to communicate with people very well. Communication is a central requirement of pretty much anything, and it's especially important in science where concepts can be extremely abstract and difficult to communicate well. What I think that means in practice is it means speak and write precisely. So always say exactly what you mean when you write and speak. And you really just have to kind of train yourself to do that. It's hard to do. don't use slang, don't use shorthand, say precisely what you mean. Don't say that a gene changes in an experiment, say the expression level of a gene changes across different conditions or something like that. It's also very important to organize your thoughts in a way that someone who's listening to you can understand what you mean. So what I mean there is provide some background about what it is that you were trying to do, say what you did, and say what you think that means, for example. Have a good, clear structure when you talk to somebody about something. One of the ways to do this is to just talk to a lot of smart people and listen to what they have to say. The listening part is especially important, but the smart people part is also important. And I want to emphasize that this doesn't mean professors and fancy scientists. Some of the smartest people I know do not have university degrees and have insights that constantly make me understand things better than I did before. So just learn how to recognize smart people. And the right way to do that is to be rigorous. So when you talk to someone, ask clear questions, define how you can be wrong when you try and do something of your own, and follow up on it when you're asking a question. So if you ask a question and what a person says doesn't make sense, ask them to clarify it. That I think is one of the number one things that people don't do, which can really help them become much more insightful. And finally, I think it's just better to have become correct than to have been correct. There's this thing that scientists do, and I suspect that everybody who's listening to this is probably pretty smart, where you learn these techniques which allow you to kind of trick another person into thinking that you were right all along, when in fact you weren't. And if you're like me, you should spend a great deal of time not doing that. Try very hard to figure out how to learn what the right answer is, even if it wasn't what you said before. Okay, so that was a kind of abstract set of recommendations, but I also have some bioinformatics specific skills. So obviously the thing that you get hired for as a bioinformaticist is the skill set that you can provide. Central to that is learning a programming language. I highly recommend R. Python is also popular. But whichever one you choose, basically just learn to do it well and go from there. Ideally, you'll learn some experimental biology as well, and if you can, you should do a little bit of it. So this could mean volunteering in a lab. It could mean ideally getting a job where you have access to people who do that sort of thing. You might just have a friend who's an experimental biologist. Talk to them and listen to what they do. Also, you're going to need to learn some probability and statistics, because a central goal of bioinformatics really is providing these statistical insights. And then finally, the last thing is gain some experience. You don't have to start somewhere fancy. And sometimes it's a great idea to volunteer for a while. So there are lots of free consortia where you can just kind of hang out. You can code something up on your own that you think is interesting and share it with others. And generally speaking, those communities will be very receptive to that. At the end of the day, what you're really trying to do is learn how to do things. So it's not just a question of sitting in a chair and making money. It's a question of being able to implement something valuable to someone else. And I think it's a good rule of thumb that if you find that whatever it is you're doing isn't providing you with more information, it's time to find something new. And then the last thing is really just relationships matter quite a bit. Make sure that the people that you work with understand what you're doing, they understand why it's important, but also you should just care about them, care about the things that they want. And that'll really help you as you progress through your career. So the last thing that I'm going to close with is just to kind of emphasize that when we think about data science, I think a lot of times people emphasize the data part and not the science part, because everyone's impressed with how many millions of samples we have and how many thousands of genes you can measure, et cetera. But at the end of the day, the thing that's really going to make you a great scientist great data scientist is to really focus on the science part. The science part is the thing that really matters to everybody. And you'll be infinitely more valuable knowing how to be a good scientist than you would be otherwise. So with that, I'd like to conclude, encourage everyone out to go out, find a problem, fix it. And thanks again to the organizers for giving me the opportunity to speak here.